All right, thank you, Manuel, and welcome back from lunch. Hopefully, we'll keep you awake. What my good colleague and friend Vefa Franchini and I would like to do is to talk about viral infections, both from the context of our research that we did together and published together, but also from the point of view of the theme of today, which is the role of veterinarians in biomedical research teams. So I'm going to start off, give a brief background to the work that we did in viral infections, and then Vefa will come up and present her research on current investigations. And then we'll finish with some comments about workforce and some aspects of biomedical research training. Of course, those of you that have talked earlier in the meeting had talked about the role of animal viruses and examples, and it's useful to actually look at the history of retroviruses as an example of a biomedical system in which we learned a lot from animal models, and we knew a lot about different viruses, especially in viral oncogenesis, that laid the groundwork for subsequent investigations in human virology, and it really wasn't until the early 80s that we knew definitively that human retroviruses were associated initially with cancer and then later, of course, with immunodeficiency disease. But you can see from this example with people like Bill Jarrett, who is a veterinarian who really understood the natural diseases and discovered feline leukemia virus in 1964, really helped set the stage for that. And I had a wonderful experience here at NIH with Bill Jarrett doing a senior postdoc in Bob Gallo's lab when we were studying ovine lentiviruses. And we were called in by Dr. Gallo to learn about the new lentiviruses, and Bill Jarrett was a senior investigator then, so it was a great experience. But I think this really sets the stage for some of the themes we have today, which is what we can continue to learn together as interdisciplinary teams. Now, the virus that Dr. Franchini works on HIV and HTLV1, but we're going to talk only about HTLV1. I got hooked on HTLV1 when I did a postdoc at the Centers for Disease Control. And at that time, it wasn't being screened for in the blood supply, and helped develop a laboratory to help some of the diagnostic testing that got me into the field, and I fell in love with it. It's a Delta retrovirus, and as mentioned earlier, it's one of those examples from the natural animal models in which we see it both in humans as well as in primates with clades, and I'll show you an example of that in a minute. In particular, we're going to focus on some of the regulation of the viruses and what we learned from both molecular systems, but also biological systems about how this virus is regulated. As mentioned, this is really if you thought of them as a group of viruses, it would be a primate T-Lymphotropic virus, larger group with various families off of that. This was from a book chapter in Fields Virology that Vefa and I published in 2007, and likely continues to grow, but it illustrates the overlap in what we can learn from the primates. Most of these were from primates in the field in Africa and other regions, but how closely they are related to the human viruses, and so they become very important in terms of comparative molecular biology, but also just in their biology in general. In terms of HTLV1, of course, it was really known that it would cause disease before really it was genomically characterized or even virologically characterized by clusters, and this is where teams of epidemiologists, both in Japan and the Caribbean basin, were categorizing diseases that they were seeing in clusters, and mostly in southern Japan, adult T-cell leukemia lymphoma was a particular subset of leukemia, presented in a variety of clinical presentations, including smoldering cases, but also a very aggressive form, which even had things like flower cells, as shown here in this blood smear. So a distinctive form of leukemia. But interesting about this was a virus that was also associated with inflammatory disease, including HAM-TSP, HTLV1-associated myelopathy, and tropical spastic paraparesis, denoting where it was discovered, but it was really the same disease, one in Japan and also then in the Caribbean. But the patients here showed not tumors, but thoracic cord inflammation, eventually resulting in demyelination. And when we first were studying this at the CDC, it was overlapping a lot with other diseases, and neurologists for a while had to really investigate how to differentiate this from other diseases, including other chronic neurodegenerative diseases. We know a fair amount about the pathogenesis of the virus. We know this is a chronic infection like all retroviruses and really needs other things to occur before we really see the full-blown episode of clonal proliferation and expansion to disease. I think a lot of the field was really focused on various aspects of, as shown here in this design pathogenesis chart, of not only the early transmission, but how is the virus regulated? How does it exist in lifelong infections? And this is where teams of investigators, from hematologists to molecular virologists to veterinarians doing animal models, really come to bear on the disease. The genome as a complex retrovirus encodes typical proteins that are structural or envelope, but also like HIV, many of the other so-called accessory genes. And you're going to hear a little bit more about that as we talk, and Dr. Franchini is going to help us understand what they're currently looking at here. So we really are presenting an intramural as well as an extramural talk. And what you'll see is really the value of both natural and experimental animal models. We also have a model that is a disease in cattle called bovine leukemia virus, which is a member of the Delta retrovirus, which is also a big problem in cattle, a natural disease. Interestingly enough, it causes a B-cell lymphoma, not a T-cell lymphoma. But in terms of its regulation, in terms of its pathogenicity and its genetic structure that causes disease is very similar to, as I mentioned, natural diseases in simians of the simian viruses, but also experimental models such as the rabbit, which is uniquely susceptible to HTLV, and also the sheep, which are a form of model for bovine leukemia viruses as well as an array of mouse models. So I'm going to turn it over to Dr. Franchini, who are going to go over some of her current laboratory foci.
What I will do today is try to explain to you how we are trying to understand which genes are important for the persistence of HTLV1. HTLV1 is a retrovirus that persists for the whole life of the host. And what is known is that the viral burden, which is a very important point in PBMC, is a predictor of disease development. So if you have more virus, you have more chances of developing leukemia or TSPM. So one of the questions that we were trying to address was to see to try to understand which one are the viral gene essential for viral infection and persisted in monocytes, because we had the sense that we had some evidence that suggested that if you infect monocytes, you can infect in vivo, and I will show you some of the evidence in the animal model. So the animal model became really very important to try to verify some of the concepts that we were deriving from in vitro studies. And we used both the rabbit as well as the macaque model. So I was asked to give you a case report as an example of a study that we have done that involves a lot of different groups with different expertise that all come together to try to now provide their own expertise for a single goal, which is to try to understand how, for example, in this case is the ORF1 of HTLV1 is important. So the laboratory that were involved in this were obviously our lab with all the postdoctoral people, the technician, as well as myself at the NCI in the intramural program, but also people that did field epidemiology that were providing samples that we could then study. And we studied 160 infected individuals from different geographical areas in the world. Also, we needed to have, of course, the collaboration of patients as well as physicians as well as nurses, because the sample come from people that have disease or not, even if they are asymptomatic still. They're followed up by the clinics all over the world. And then we needed a core support that would actually perform clinical assay to analyze seroconversion, actually to document actual infection, and also to measure the level of viral replication in any given individuals. And all of this collaborated with each other, and the results that we were obtained allowed us to formulate hypotheses, which then we could test in the animal model to see whether the hypothesis was correct or not. And this, of course, involves IACUC, regulatory committees, as well as animal husbandry and animal care personnel, veterinarian scientists. So as I was telling you, I will just briefly summarize some of the results that we had on studying the ORF1 of HTV1, because it's a very interesting protein, very small protein. So retroviruses are very special because they can use very little genetic material and to do a lot of things. So often proteins are multifunctional and they are able to do different things. So in the case of the ORF1, we know, I can just summarize for you that we know that it is very important for viral transmission and persistence in vivo. And this is, what is really key, as I said at the beginning, is that the virus, by expressing the P12 and the P8, is able now to infect monocytes. And without the infection of monocytes, you don't really infect in vivo. So the monocyte appears to be the cells that are able to get infected and maintain the virus in vivo, perhaps by transmitting that to the T cells. And this is probably due to the ability of these cells to migrate in the tissue, to present antigens, and of course, you know, the antigen is presented to T cells, and the T cells are indeed some of the target of HTV1 infection. So the ORF1 is a very interesting protein. As I told you, basically it's only 99 amino acid. But what is happening is that, you know, from the ORF1 you can generate by translation a protein of 12 kilodalton, which then gets cleaved, and that gets cleaved in, and the cleavage eliminates this ER retention signal, which is a non-canonical signal, which then allows a second cleavage in here, and then you create another protein which is called P8. You can see in here that, you know, if you have, this is the uncleaved precursor, which can be cleaved in the P8. And what we found, which was pretty interesting when we started to do reverse genetic in patients infected with HTV1, is that some patients did have mainly the precursor, some patients did have mainly the P8, and some patients had both. So we became interested since we knew that these two proteins have very different effect on T cells. Remember, this is a virus that infects mainly T cells, CD4 and CD8 cells, as well as monocytes. So the P12, so the uncleaved precursor, was able to increase T cell proliferation by increasing STAT5 and then FACT activation, as was demonstrated by our laboratory as well as Dr. Lairmore laboratory. But also, the P12 was able to decrease MHC class 1 surface expression, so clearly trying to make perhaps these cells resistant to the cytotoxic activity of the CD8 cells in vivo. And was also able to downmodulate ICAM1 and ICAM2, and which are important for increasing the strength of the immunological synapse, which is the point of encounter between cytotoxic T cells and the infected cells. And this could also lead to elimination or resistance of the infected cells, not only probably from CTL, as I will briefly summarize afterward, but also from antibody-mediated cytotoxic activity mediated by NK cells. So instead, when we look at the P8 function, and this was done all using, you know, overexpression system, using the cDNA from the virus, what we saw that the P8 had the opposite effect. And for a little while, we couldn't really understand what it meant. So what the P8 does is, instead of increasing T cell proliferation, is actually downregulating the T cell receptor signaling, and therefore is decreasing T cell activation. So it's the opposite effect than P12. But also, we found that the protein is made, goes to the cell surface because it's cleaved, and then is able to be transferred through tunneling nanotubes to neighboring cells. So all of a sudden, the virus is able to make a protein that is able to invade also uninfected cells. And that's an important point, because if you now invade a CTL or a cytotoxic T cells, and you downregulate the TCR, the T cell receptor, then these cells will be unable probably to kill the target cells. So it's a very good way to, you know, to basically prevent CTL activity of the CD8 cells. And in addition to this, this protein, by going to different cells, was also able to improve, to increase the transmission of the virus from one cell to the other. And I think I have here a video that I would like to show. So you can see this is a live imaging. This is one cell. In yellow, you see the virus, which is labeled with an HTV1 GAG YFP, which was generated by the late David Darcy. And in red is the P8 protein, which is labeled with the cherry. It's just so you can detect them easily. So you can see in here, and what I will show you is that this cell moves continuously as a T cell and is making all these tunneling nanotubes, which are filaments of variable size and length, and they reach other cells. So I can try to see. Oops, I don't know if it didn't work. Oh no, I don't know why it didn't work. It did work before. Yeah, let me try again. Yes, it does. Sorry. You can see near all these filaments. You see the yellow going to the next cells in there. You can see the filament going there in this direction and in this direction. And basically, all of this occurs within minutes. So within minutes, we can see the transmission of the virus as well as the P8 protein. And we have a lot of evidence that shows that this is indeed is all documented biochemically and so forth. So, okay. There we go. Okay, so then we were interested to see whether the two proteins together were needed and how in vivo, and how this in patients infected with HTV1, and how the phenotype that you get expressing the ORF1, so either a phenotype that expresses both proteins, as I showed you before, or mainly the P8 or mainly the P12, could actually affect virus load. And when we did this analysis on 160 infected individuals, you can see in here that we see that if you patients that express both the P8 and the P12 actually had a higher virus load than patients that express either one or the other, suggesting that the production of both proteins is probably optimal for the virus. But this is, again, is an interesting observation, but is only a correlation. Is that really true? So to try to prove it, what we did is we generated a molecular clone which had point mutation in the cleavage site that would lead either to the production mainly of P12. This is in black here. This is just the quantitation of the amount of protein produced by any given cDNA isolated from the patient, or both, as you can see in here, or only the P8. So we isolated these clones. We basically, the name is not important. I will take you through this afterward, but clones that express both P12 and P8, some that express only the P8, one that express only the P12. And of course, we use a clone that expressed neither as a control. And then in this way, we created cell lines, which then we could, they could make virus, equivalent amount of virus, and then we could irradiate and then inoculate animals with the different cell lines to try to ask the question, how do these viruses fare in vivo? Are they equally fit or not? So, but before doing that, we looked at the infection of dendritic cells, and you can see in here that if you have no ORF1, you get no infection of primary human dendritic cells. And if you now take the THP1 cell lines, which is a human monocytoid cell line, and you ask the same question, you can see that again, if you have no ORF1, you get no infection, while instead if you express both, you do have a nice expression. If you express only the P8 and not the P12, you still have infection of these cells, but if you now express only the P12, you don't. So this is suggesting to you that the P8 for infection of monocytes is very important. So then we went to try to do non-human experiments both in rabbit and non-human primates. The reason why we tested both is because the rabbit is a very, as Dr. Lairmore told you before, is a very sensitive model. You can get very good infection, but the non-human primate is a natural model of infection because they are infected by HTLV1, which is very, very similar to HTV1. So we were wondering whether one model could be better than the other, or whether we will get different kind of answer using different models. So this is an example of what happened is that if you take the rabbit model, you can see here that whether you use the wild type, here we are looking at seroconversion. So this is the typical blot, Western blot that you can do. It's still done on HTV1 infected individual as well. You can see all these bands that really correspond to a variety of HTV1 protein. So all animals seroconverted when inoculated, irradiated cells expressing the wild type virus, which express both P12 and P8. But when we inoculated the knockout virus, we also got very good infection. So when we looked though at macaques, it was really surprising because we saw that there was no infection at all. when we had no P12, no ORF1 in the molecular clone of HTLV, suggesting that the two models, the readout in the two models is quite different. And I'll explain to you also afterward what happened. I don't know why this slide is in here. Oh, there are a bunch of slides that are missing. Anyway, I'll summarize it for you. So then what happened is that when we did take the molecular clone that expressed only the P8 or the P12 alone, either the P8 only or the P12, basically we demonstrated that you needed actually both to get optimal infection of non-human primate. And so this, again, indicated to us that the results that we obtained in humans was actually mirroring what we were finding in non-human primate, but not what we did find in rabbits. So I think that this data suggests to us that the rabbit model has value, but is not giving us the same kind of answer. Thank you.
So yeah, I think this illustrates also what we had talked about earlier, is the finding the right model, finding the right systems. I was very fortunate while I was at Ohio State to lead a program project grant in which we were looking at lymphocyte transformation and disease. HTLV was one of those models that we used, and this was a collaboration that involved 5 projects and 3 cores. 2 of the projects were at Washington University with Lee Ratner and Kathy Wellbacher, as shown, both MDs, and this was a good illustration of our theme today for really the role of transdisciplinary teams in research. As Vefa mentioned, when we looked at the rabbit model and looked at the whole virus and looked at one of the convenient parts about doing a model like that is to be able to do more interesting studies in terms of where is the virus, and doing things like where is the virus, and asking questions like, is the gut important? And one of the things we found in that model published in Journal of Virology is that the gut is actually a very potent source for the virus in terms of where do we find the virus during the chronic infection in the rabbit model. Not too surprising for those of you who work on HIV, that we found that especially early on, the virus is increasing expression, but later stages accumulates within the mesenteric lymph nodes. And there's a lot of data I'm not showing to support this, but showing the value of the model in terms of some pathogenesis studies. And this would allow us to do things like what would happen if we immunosuppress the animals with cyclosporin at different stages of the disease, showing that T-cell activation was very important. If you block that by early cyclosporin treatment, you didn't get the infection, indicating that this virus needs to get in, activate T-cells, and really spread. But after that, may be resistant to that. So rabbits infected a week prior to cyclosporin treatment really didn't develop the same kind of viral loads as those that were developed afterwards. And again, this allows us to do kinetic studies following infection, and then different targeted suppression of that. So that's one of the values of that model. Now, switching gears in terms of what are some things we learned in context to this workshop today, and I'd like to go back and mention a couple of things that I've been involved in more recently as I went to the dark side to become a dean at UC Davis. Thinking about things like developing the next veterinary workforce in research. And we were fortunate to have the NRC study come out on veterinary workforce. Some of the interesting recommendations which relate to what we've talked about here is the need for academia to increase commitment to research and funding, and really developing future faculty by working across disciplines and across professional boundaries. I think this workshop really demonstrates that quite conclusively. But also, as we look across the globe in terms of our security, food is going to continue to be important, and veterinarians trained at multiple junctures of that are going to be important, but also engaged in interdisciplinary and One Health solutions. So that was actually brought out within that workforce. Sue Vanderwood and Jim Fox and I and others were involved in a physician-scientist workforce working group as part of an NIH study to look at physician scientists. And you're going to hear more about that, but just a couple of the highlights of that in terms of that, which came out in 2012. And we came back and lobbied it in the Capitol Hill, organized by the AAVMC, to try to increase funding for NIH. Manuel, you're welcome. We were lobbying for you to increase your funding. But it really came out of the fact that if you look at the data, it's really apparent that if we can fund physician scientists And what we mean by that is both MDs, dentists, as well as veterinarians, there is a good outcome. So why would we be talking here about why veterinary scientists are needed? For one, I think it's pretty clear to this audience, and as demonstrated earlier today, that there's unique contributions. Specialized training in animal biology and medicine and modeling of what is the right models, how is that we're at ease going across species and understanding the differences. And contributing to that really helps to better animal models. But also, as we've seen, things like emerging pandemics and epidemics really are prominent examples of why we need veterinarians in the workforce. We only have 28 veterinary schools, 2 more are being added, so we maybe have 30 here within a few years. But not all of them train veterinary scientists. At least 19 offer some sort of dual degree training. We have about 17 programs that offer some postdoctoral T32 or other types of training, either post or pre-doc. T35s are out there. What we do not have is veterinary scientist training programs dedicated to dual degree students. We do have some slots in the MST pre-programs, but that's going to be important here as you see some of the data. So one of the things that are challenging us in this is recruitment. Many of the students come to us are clinically focused, about 80% of those, and we may be our own worst enemy at we are recruiting, and so I think we need to pay attention to the pipeline coming in, but also training and mentors. That was mentioned several times today, of putting them in context to the teams they need to have that training. But also, we still have to face up to the fact that their starting salaries and their student debt ratios are contributing to them making decisions not to go into research. And so that needs to be dealt with. And there are some glimmers of hope with new loan repayment programs in which veterinarians are being eligible for. But that's something that was recognized by the working group. And certainly the mechanisms for loan repayment and training grants are clearly important. Now, how do they compete? Well, they do well. Once they are trained, they compete very well, or equivalent rates as physician scientists. So veterinary scientists are trained, and they're worth the investment. If you look at the ones with K awards, they do have a good ability to go on to get R awards up in the 70% range. So suggesting that we do have a good outcome if we are to invest in them. So why are they needed? Well, we only need to look at emerging viral diseases to see, because of the amount of zoonotic diseases, that about 70% of human pathogens are zoonotic. So this One Health approach is really integrated within that. So on our zoonotic and emerging diseases, we see as a map here that shows either emerging diseases or re-emerging diseases. We're going to continue to face this as a challenge. So just a couple of quick examples at UC Davis. We are there, our mission is quite simple: to advance the health of animals, people, and the environment. So we've incorporated the One Health theme within the entire mission of the school, and this goes from surgeons to epidemiologists. And really, we are there to address societal needs using this One Health approach. And what we see it as is, as you've seen, this interface, this Venn diagram, but also involves a lot of different fields like biology, ecology, as mentioned this morning, but also engineering, earth sciences. All of these involve really transdisciplinary approaches. And the drivers of what our problems will continue to be here. Climate change, problems are going to continue to emerge, and it's only through this kind of One Health approach that we're going to be able to tackle this. So one of the things we've done is to form a One Health Institute, which started from a small donation of a wildlife health center about 10 years ago, and now has over a $20 million annual budget. And this is a group that really is focused on working at that interface between animals, people, and the environment. Jonna Mazette leads that. She's a member of the Institute of Medicine and is a fantastic leader. As mentioned earlier, USAID is really a major portion of their funds. Predict2 was just funded to look at emerging pandemics. And what they're doing is really at the interface, more on the viral diagnostic side, but using platforms in hotspots throughout the world to predict the next emerging pandemics. Clearly a place in which these transdisciplinary teams are needed, and we only need to look at Ebola to figure out why. And as mentioned earlier, it's really a capacity training as well. This gives some of the data from their first 5 years. And this is, I think, really important for the workforce to be out in around the globe to build capacity, but also in their discovery, creating new knowledge about the viruses, as mentioned several times, in the field, where we're learning a lot more about the natural reservoirs of the virus and also human pathogens at this interface. And this shows you an example of some of their trapping that they're doing in the field as they build up capacity and train in the field. We're taking this back into the classroom. This is an example of a hybrid learning interface in which we take those examples back into both undergraduate and graduate courses, and we feel that that's a very important part of that, is how it comes back to the universities, back into the training of the next generation of One Health practitioners. It was mentioned earlier about how do we get more medical students involved. One way we found effective is to have One Health clinics. This is an example in Knights Landing, which is only about 25 miles away from the university, but it's a predominantly Hispanic population, immigrant workers who have animals and a lot of unmet health needs. And what we do is we have both the medical students and the veterinary students together. The veterinary students are conducting the animal clinic, the medical students are seeing the patients, and then they meet for joint rounds. And it's pretty incredible to see the students when they start to work together like this. And I think that that's a potential model that many universities are taking early on in the education of medical students that I think is going to be very helpful. The primate centers, as mentioned, are going to be very critical for that, especially on the experimental side for really the precious 7 primate centers that we have in terms of some of those resources. And these need to be linked with our other resources in cancer, in our CTSCs as well, to really reach the power of that. Other centers, such as the Center for Comparative Medicine, illustrate focus on mouse models, actually, primarily. And we have the mouse biology program as well, led by Kent Lloyd. And I think the combination of having multiple model systems with dedicated scientists is very powerful in terms of this One Health approach. We take advantage of our CTSC, our CTSA locally, and several of our universities are now linking that have CTSAs, and I want to continue to hopefully encourage that, because I think that's a way also that we can leverage our resources across and also become more synergistic. An example of that is in training. This is an example of scholars that have been trained by our CTSC, and we have representatives here of our CTSC, which we've become very important partners with, and our faculty that have trained in those programs come back to be leaders. Michael Kent is a leader of our Center for Companion Animal Health, but also is involved in R01 level funding now with Bill Murphy in drug discovery and immunotherapy against cancers. So a very strong example of taking advantage of CTSC training for our faculty. I mentioned engineering. I think that's another area that comes to bear, especially when we have problems related to things such as surgery, three-dimensional recreations preparing for surgery, combinations of using bone matrix with medicine, bioengineering, and our craniofacial surgeons have really led the way for new approaches to bone replacement as well. So engineering, as mentioned, the cancer centers and the link with the consortiums are very important, and I was glad to see that continue to be emphasized. This is very critical to our university, like many of the others that have comprehensive cancer centers. We, like others, have created a Center for Clinical Trials. I think this is a way to have, as mentioned, parallel clinical trials at the universities for our patients, but also in our that are parallel and work hand in glove with our other clinical trials going on in our medical schools. But you can see it's not only cancer, but a variety of other diseases that are important to humans that the trials are going on, including ophthalmology and others in terms of novel approaches, both in cancer, but also chronic inflammatory diseases. The other thing that was mentioned earlier is, as well, which we feel very committed to, is innovation and entrepreneurship. And we've created an academy with engineering, medicine, and now biological sciences to put graduate students, postdocs, and faculty together who have platforms and allow them the training and mixing with venture capitalists and others to really get that entrepreneurship further along in the stage to allow that to occur. The graduate programs are going to continue to be very important, and as as we look at our interdisciplinary graduate programs in particular, we see a lot of One Health themes related to that. These are 4 or 5 examples in our university. And I just wanted to finish by showing some highlight. Kim Dodd was a dual degree student in our VSTP program, which is funded from endowments currently. She received her PhD at CDC, was especially trained, and during her last year of veterinary school was called to Sierra Leone to help in that effort. And the man pictured below is Dr. Brian Bird, who was an alumni at the CDC, again, trained through our VSTP program. So these VSTP students are out there in the world and on the front lines, and I want to see that encouraged as well, and I think our discussions of that dual degree training is really going to be critical in the future. So thank you very much. Do you have questions for Dr. Franchini or I? Yes.
Hi, Bernadette Dunham with the FDA. I think that's been a fantastic capturing of everything we really want to embrace. I think the whole idea of today's session into tomorrow is how do we tell the story? And you captured that beautifully, and I hope that we could see our counterparts in some of our medical schools do the same. We recently interacted with a number of starting programs where they'll have a veterinary student along with a medical student work together and share what you've just highlighted, some of the benefits and the stories, and really understanding how it can be One Health. But it's getting this into what you said earlier, how do we communicate and enhance everybody to be aware of the advantages of working together instead of in silos. When it comes to the grants, we're here at NIH, you've actually really highlighted the success story of so many acquiring the grants, because many times they think there's a disconnect. And we heard earlier, even study sessions, do they know and embrace when you do want to have both collaborating? Can the deans recognize that when it comes to promotion of universities for their professors? And you've highlighted beautifully how that can happen. So I hope everybody will take advantage of what you've shown here and get the message out a little bit more, because this would make it a real One Health win. Thank you.
Yeah, thank you. I think the communication is absolutely critical. I think at the leadership level is very important. We have a lot of leaders here of centers, and I think that's a very important group. At our own universities, the health sciences team meet frequently, and we're constantly talking about it back and forth. They actually embrace us, which is very important too. That alignment is critical. Department chairs, center directors, so absolutely. And we found that early on with that medical student interface, we bring an entire class together around case studies, much like what we're hearing today, and they work together. Of course, the medical students are really concentrating on the patients, but we use cases like toxoplasmosis and as case studies, which really illustrate that connectedness back and forth. So thank you for those comments. Yes.
Yeah, Mike, just to follow up on your comments too. I'm Nick Kenyon. I'm at UC Davis also. I'm pulmonary critical care, and I've been the head of our TL1 program at the CTSC for the last 8 years. So just to reiterate what Mike was saying, we've had at least 1 to 2 DVM PhDs in our training program every year with MD PhDs and MD students. I've also been in the same training program at the junior faculty level 10 years ago with Michael Kent, and there's been a number of folks that I've interacted with both on grants and education projects since that time. I've also had vet critical care residents in my lab now, and if I'd say there's one area that we could probably improve, it's probably the training and the interactions at the resident and fellow level at the vet school. Obviously, there's more hours restrictions put in place for medical school, for medical residents and fellows, so they do a little bit more research. I can say the vet critical care residents work harder, and they have very little time.
Yeah, one thing we run into there is that when they're taken out of the clinic, our faculty that get on the training grants are then the first thing the hospital director says is, how are we going to cover the patient load? Because we have 50,000 patients coming in, how do I do that? So what we're trying to look for is offsets of money, endowments, other ways that we can hire staff to come in and replace them during that critical period of time. But it is a very, very much of a challenge for us. Yes.
So, Mike, thanks very much. Francisca Guido. I'm going to echo what the 2 previous speakers said. You, of course, demonstrated a beautiful example of like what's being done and how it's done well. My major question or concern is, how is that transported to other places? So it goes back to communication, but it's more than communication. It's kind of, how is it cloned?
Mm-hmm. Well, I think Debbie Kachiva can comment on this, but one of the things that we're doing at the AAVMC level, so that's all the veterinary medical schools, we had that theme of One Health 2 years ago, and that came out of they have a standing One Health committee now within that group, and I think Debbie's done a great job of organizing our, what we call the COHA group around the CTSCs, but I think we're learning a lot in those consortium efforts as well, trying to leverage that. I don't know where, Debbie, you want to make a comment or not?
We'll have a chance to, Debbie Kochever-Tufts, we'll have a chance to talk about that a little bit more this afternoon, but I do think trying to form consortia around some defined goals helps to make the One Health discussion a little less diffuse, because I think as we've seen today, there's a lot of great things going on, but it becomes all-encompassing, and so you really have to try to focus in, and so the CTSAs have sort of done that for us, because they have their own priorities, and now we're, as the 10 vet schools that have CTSA institutions, can really focus our efforts to try to drive those priorities, and hence, we hope, gain some resources that will help all of the vet schools collaborate more effectively.
Yes.
So I hear a lot of great, and your presentation was fabulous, but I hear a lot of great talk about how veterinary schools are getting involved and the programs that we're supporting to make the One Health initiative stronger. Do we know, is the same kind of conversation happening on the medical side? Are we talking to the medical community and med students and how we can create the same things to connect the two communities together?
Well, I think it's earlier, I think we heard that from Carolyn, we're sometimes preaching to ourselves. But what we're trying to do, and I think this is clear in the COHA group, we're trying to now take that conversation directly to our leaders of our CTSC. I presented to the EAB of the CTSC just the other day, about 3 weeks ago. And we find the audience to be very receptive, but we need to be very, put our foot in the door and keep that door open at all times. And I think when we look across the other schools, I see that. They're done in different ways. Some are at the student level, some are at the faculty level. So I do see much more momentum than it was, let's say, 3 to 5 years ago, where we're not doing websites as much as we're actually doing projects. And there's a difference between talking about definitions and actually doing the work and getting it funded. And you're right, the next level is to really force that message into, and not force it, but gently persuade. Yes, Debbie?
I was just going to add, some of you probably attended Zoobiquity conferences nationally, of which there's probably been 4, maybe 5 now. And because Boston has such a concentration of medical schools, there's 4 med schools and 1 vet school, we're going to have a Zoobiquity Boston at the end of April. And we tried really hard to get all the medical students invited to events so that we can get the medical students, the house officers, the folks that were the sort of the next generation of this, thinking in terms of this now, not 20 years into their career. And so I hope we've heard that from everyone today, that that's a target. And I do think there are things happening on that front.
Yeah. We out of time.